Recording begins. This is October 15th, 2025, Biology 105. Hello, if you're watching the recording. Okay, here we are. We're in week 8, right? We're going to be covering photosynthesis starting today. So we're kind of like almost caught back up with our schedule, which is pretty exciting. It's pretty cool. This is a big week, so I'm just making it. I usually only do one of these announcements each week, but I just want to make sure everybody's on top of these things, right? You've got that online lab midterm, okay? It is due by the end of the day on Friday. You got to have it done by the end of the day Friday. You've also got a photosynthesis graphing assignment / pigment identification assignment that is associated with your lab this week. The lab midterm does not cover this week's lab. It covers weeks 1 through 7, so you can do it anytime this week. All right, you already have the materials. There's also a reading quiz on chapter 10, which covers photosynthesis. That's also due on Sunday. And so, yeah, there's 3 assignments this week instead of the usual 2. Good deal. Any questions before we, also, sorry, yeah, next week's a regular old week. No reading quiz to try to make life a little lighter, just a lab assignment. And the week after that, you've got your second exam, exam 2, on the Monday before Halloween. It's a spooky exam. All right? Cool. It's not cumulative. It just covers material since exam 1. The study guide's already posted. Just review the study guide. Question? Yes. But there's a final exam study guide that lays out every topic you need to know from all the previous exams. Yeah. Cool. And then, yeah, it'll be week 10, week 11, week 12, week 13, fall break. Exam 3 start before fall break. Then we come back and we have 2 weeks, and then we have our final. Sounds great, right? All right. It'll be here before we know it. Let's go ahead and just, here's the link for the online lab midterm. It's the start of this week's module on Canvas. Let's go ahead and pop open that respiration PowerPoint. Here's my photosynthesis PowerPoint, so I can easily switch over to it. Okay. Enable the editing. Here we go. So we made it through our chemiosmosis slide on Monday. So remember, you have these electrons that are at a high energy state. They were at a high energy state in glucose. You rip them off, all right, of glucose through glycolysis and pyruvate oxidation plus the citric acid cycle. So stage 1 right here, glycolysis. Stage 2, pyruvate oxidation and the citric acid cycle. This breaks down the glucose, gives you a tiny bit of ATP via substrate level phosphorylation, breaks the molecule down so it releases CO2. All of the energy at that point is held in these reduced electron carriers. They are reduced because they're carrying electrons. Those electrons are at a very high energy state. They are then going to be fed into the electron transport chain, where they fall from a high to a low energy state. Their lowest energy state is going to be when they land on oxygen, okay, the terminal electron acceptor in this process. As they fall to a lower energy state, the energy released is used to pump protons, generating a proton gradient. And this is actually called the proton motive force. And then those protons are forced to flow through ATP synthase, which acts like a dam. And as these protons flow through it, diffuse through it, if you wanted to say, all right, the energy is going to be used to generate ATP. So you start with the energy in potential energy on the glucose molecule. Potential energy on the glucose molecule, you convert it into reducing potential, which is just another potential energy, okay? In that it's all about the position of electrons. Then you convert the energy into a concentration gradient, a proton gradient across the inner mitochondrial membrane. and then you harvest that gradient, that concentration gradient's energy, in order to generate ATP, which is now another chemical potential energy. All right, a lot of energy transformations going on here. Question, please. Because of the gradient, yep. Exactly. Yep, they want to move in this direction, but they can only go through ATP synthase. Yeah, solid, no problem. All righty, I love it. And then we looked at the electron transport chain in more detail, and then we looked at the where I mentioned, hey, listen, for an electron to go from me to the table must be more electronegative than me. Otherwise, the electron wouldn't go in that direction. We cool on that? Awesome. We'll talk a little bit more about oxygen, but remember, it's the most electronegative component of this, and it's going to serve as the terminal electron acceptor. Once the electron goes on to oxygen, you get some water, and that molecule's pretty darn stable. Awesome. Proton motive force, chemiosmosis. Remember, the overall process is called oxidative phosphorylation, but oxidative phosphorylation is the combination of chemiosmosis and the electron transport chain. So I'm going to actually write that down. Oxidative phosphorylation, all right, is the umbrella term. That's an umbrella. There's a little band on it. And it includes the electron transport chain and chemiosmosis. The electron transport chain pumps the protons, generating the proton motive force, and chemiosmosis harvests the proton motive force, generating ATP. All right, and oxidative phosphorylation, that's that umbrella term for the process that generates the vast majority of ATP in cellular respiration, CR. Yes. You know, when you make it bigger, it's almost legible. Questions about that? Yes? Proton motive force. So right there on the slide at the very bottom, it's the proton gradient. So if you look at this image here, which I'm going to put onto the actual main projector because there's a video. I want the video people to see the same thing. This is the proton motive force is the concentration gradient across the inner mitochondrial membrane. Solid? All right. Cool. So yeah, I really just want to make sure everybody's comfortable with the terminology because it's really easy to get chemiosmosis and oxidative phosphorylation mixed up. I still interchange those terms sometimes. Yeah, you kind of have to remember it. I mean, you could remember that that oxidative phosphorylation includes both the redox reactions of the electron transport chain, so it's oxidative, and the phosphorylating step of chemiosmosis, where the ATP actually gets generated, if you really want to, try to keep it all in your brain. Solid. Let's keep on moving forward. I don't want to squander our forward momentum. We're about to get into photosynthesis in week 8. That never happens. All right? So if the electron transport chain doesn't work, or if the inner mitochondrial membrane is permeable to protons, you die. You can't make ATP, you perish. Okay? So this is meant to illustrate to you the importance of this process, and also how both the electron transport chain and the actual impermeability of the membrane to protons are essential. Okay? So, like cyanide acts by blocking one of the members of the electron transport chain, preventing it from dropping off its electron or taking up a new one. And if that happens, then the entire electron transport chain just backs up. It all stays reduced. And so you can't keep pumping protons. This poison here, DNP, I forget what it stands for, it makes membranes permeable to protons. And if that happens, then you can't generate the proton motive force, you can't make ATP, you perish. Cool? Sick. Here's a summary figure. You've seen this figure a bunch of times, and every time you see it, there's a little bit more information. Now it's got numbers associated with the amount of ATP. Remember, I'm not asking you to memorize any of these numbers about how many NADHs or FADH2s get generated by each step, because this is an estimate. This is like the ideal, amount of ATP that could be yielded and ideal amount of reduced electron carriers. But I want you to know where the majority of ATP comes from. I also want you to know where the CO2 gets produced. I want you to know where the oxygen gets consumed. All right? And I want you to know where the energy's at following each stage of cellular respiration. Those are the big takeaways. All right? Any questions about any of those points? All righty. Sometimes there's no oxygen. And if there's no oxygen, you know what happens? The electron transport chain backs up. And if the electron transport chain backs up, you can't pump protons. And if you can't pump protons, then you can't make ATP. So there are ways to survive periods of anoxia, all right? Periods of oxygen deprivation. And one of them is fermentation. So we carry out fermentation in our muscle tissue, all right? When your muscle cells are working a lot, making a lot of ATP, okay? You burn through all the available oxygen, and your muscles start to undergo fermentation, or the cells surrounding your muscles start to undergo fermentation in order to generate ATP, and you end up producing a bunch of lactic acid. And that lactic acid can lead to soreness. All right, you've heard about this at some point probably, like lactic acid buildup and all this. But anyways, that's fermentation. Also, alcohol is generated via fermentation. Okay, so what you're doing is you are taking cells that normally would carry out oxidative phosphorylation, and you're preventing them from being able to carry that out because there's no terminal electron acceptor anymore. So the electron transport chain backs up. Cool? Awesome. So how do you get some ATP so you can survive? Well, you just carry out glycolysis over and over and over and get a little bit of ATP via substrate level phosphorylation. And sometimes it'll be enough for cells to survive. Yeasts, bacteria, many of them can survive off of that ATP yield. Okay? You as an organism cannot survive on fermentation alone. That's why we can't just go underwater indefinitely. All right? So what allows glycolysis to continue running forward? It's a little bit counterintuitive, okay? Because we've been talking about these electron carriers, and when those electron carriers are reduced, when they're in the NADH form, that's good because they have energy now. But here's something, okay? In order to break down glucose, you have to pull an electron off of it. A couple electrons off of it, actually. All right? Glycolysis is the breakdown of glucose, and it yields a little bit of ATP. But how are we pulling those electrons off of glucose? Well, that's actually with NAD+. So the real problem here in an anoxic condition is, check this out, the NADH can't reduce anything. So you've got no NAD+. And if you've got no NAD+, you can't carry out glycolysis. So In fermentation, the additional step of fermentation is you take NADH and you reduce the pyruvate, all right? The product of fermentation. So you throw electrons onto it in order to desperately generate more NAD+. And here's the problem. That doesn't give you something useful, okay? In fact, in both of these examples, it gives you something poisonous. So that makes it a less than desirable process from the perspective of the organism. The organism would prefer to undergo oxidative phosphorylation and carry out a full, get all the energy out of glucose that it could have gotten. Because there's a ton of energy left in this pyruvate molecule. Now, why can't it just use the lactate? And the answer is, it can in some ways. It's super complicated. This pathway is really complex. But the foundational concept is you need NADH in order to oxidize glucose, okay? And you need NAD plus in order to oxidize glucose. And the step here in fermentation of regenerating it turns pyruvate into a toxic product. So you get way less energy than you would have yielded previously. But it's a survival strategy, all right? So that's fermentation. And the reason that I bring it up is because understanding this requires a pretty thorough understanding of all of these concepts of reductions, oxidations, how the electron transport chain can get backed up, oxygen's role in the electron transport chain, the proton motive force, all of these things. This is a great, one step beyond that requires understanding of foundational concepts. Questions? Yes, please. I'll get you next. I see you there. Yeah.
The regeneration of NAD plus with pyruvate is poisonous?
This product here, lactate, is poisonous. So NADH throws its electrons onto pyruvate, and it either generates lactate in lactic acid fermentation or ethanol in alcohol fermentation.
Those byproducts are poisonous.
Exactly. They're both poisonous to the organism. If they get to too high of a level, then they'll kill the organism.
Okay, so an organism could do fermentation for a little bit, but they don't want to do it for a long time.
Yeah, or bacteria can do fermentation for a long time if they keep moving if they don't become saturated by the ethanol, because they exude the ethanol, right? So when you capitalize on bacteria or yeast to produce alcohol for human consumption, basically what you do is you put them in a tank with a bunch of sugar and you prevent oxygen from entering. And they'll just start to carry out fermentation desperately. But eventually, the alcohol concentration gets so high that the yeast or the bacteria dies. It kills itself because it's poisoned by the ethanol. Now, they've made some GM yeasts already that can withstand higher concentrations of ethanol, so that you can have a fermented product that's more concentrated in alcohol. But really, you can just distill or do, what's fun? Here's a fun thing for fun. You ever hear, I grew up in a very small town, and we drank ice beer. You ever drink an ice beer, like Natty Ice or something like that? So that ice, that actually means something. It's a way of concentrating alcohol in a solution. What you do is you cool it until the water starts to crystallize, and then you filter out the ice. And now, without boiling the beverage, you've concentrated the alcohol because the alcohol didn't freeze. So, you get a higher concentration beer that you didn't have to what's the word? You didn't have to add alcohol to. What's the word for that? Fortify. Like a fortified wine is when you just pour ethanol into wine. I got one ahead of you. Question?
I was wondering if you could go back to the fermentation cycle.
No, I totally can. Which one? This one?
Yeah.
Yeah, no problem.
Thank you.
Yeah, you're welcome. A question?
Just by curiosity, I want to know where you're from, because when you tell these stories.
Oh, yeah. It's a town called Roslyn, Washington. The population was 930 people. It used to be a coal mining town. And when I grew up, the only business that was open past 8 was the post office. And so we as kids would go play hacky sack in the post office because they had lights on and a heater. It snowed. That's my childhood. Yeah. And we had a class in school called science where we were graded on building a catapult and how accurate the catapult was. And that was my education before I came to university at Humboldt State. Yeah. There's just a little bit of fun trivia about me. Thank you for demonstrating an interest. I find myself fascinating. Cool. Was that enough vamping time for you to get the notes from the fermentation slide? Additional questions? Yeah.
So when would lactic acid over alcohol fermentation be performed? Like, is there specific conditions where one is chosen over the other?
It's different. It's an organism by organism thing. I am positive that there are examples of organisms that carry out both. I don't know one off the top of my head, but usually it's organism by organism. Like we don't carry out ethanol fermentation, thankfully, because if you did, you would be, you'd work out and you'd get drunk as you worked,
So
We only do lactic acid fermentation, exactly. Awesome. Yes.
I think I read an article. It was like some guy that couldn't stop getting drunk because when he would eat anything sugary, he had like this rare thing called auto-brewery syndrome, where like his stomach would like convert sugars into alcohol and carry out alcohol fermentation.
That's like a nightmare. That's just an absolute nightmare of you. Now you can't have anything that has sugars in it because you can't avoid sugars fully. It's nearly impossible. Yeah, that's wild. I love it. Anybody else? No? Okay. Yes, question.
2 4 1 slide.
Yeah.
Just to check. So in the example of pyruvate to lactic acid,
Yeah.
NADH would be a reducing agent.
This is so great. I'm so happy you bring this up. It's a great chance to review that terminology. I'm going to give you 4 John points. Well done. All right. NADH is the reducing agent. It is reducing pyruvate to lactate. In this process, NADH is oxidized into NAD+. Therefore, pyruvate is acting as the oxidizing agent. And we have to assume that pyruvate has a higher electronegativity than NADH. So this figure actually tells us a lot if we remember all of our little details. Solid. Anybody got any other questions about the satisfying answer? Nice. All right. So another thing that's important to bring up. We always teach cellular respiration as if glucose is the be-all, end-all. As if glucose is the only thing you could eat that would drive cellular respiration. But that's not the case. You probably know somebody who's gone on a low-carb or a no-carb diet, for example, or just, cut sugars out, and they live, if you call that living. Oh, yeah, we have fun. We have fun here. All right. So other molecules, other macromolecules can be fed into cellular respiration. There's usually just another step. Like, think about a fat. You break a fat down, a triglyceride, and you're going to get some fatty acid chains, which are hydrocarbon chains, right? And you're going to get some glycerol, which is basically a sugar. So you break down a triglyceride, and you can put that glycerol right here into this G3P molecule, which is an intermediate between glucose and pyruvate. It's got a phosphate on it already, so like substrate level phosphorylation is going to work. Blah, blah, blah. There's so many details to all this. But the point is, look, carbs, fats, proteins, they can all be fed into the process of cellular respiration. So they can all be harvested for energy. It's just that glucose is the molecule that goes through the maximum number of steps and yields the most ATP. So it's the best sort of teaching model, all right? Because the other ones just run through fewer steps. Solid? Yes, question. And then I'll get you next, yeah.
So, amino acids, for example, can go straight to the citric acid cycle?
Yes.
Is that what it's showing? Okay. So
Yeah.
So fats and proteins can skip some of the steps.
It's more accurate that the breakdown products of an amino acid can be fed into different stages of glycolysis. And
Sugars can only go through that one.
well, sugars start here at the beginning, and the more steps you go through, the more energy you get. Yep. If you just get jammed right into the citric acid cycle, you're not getting any of that glycolysis energy or any of the NADH that came from stage 1 or from pyruvate oxidation. Yeah, of course, yes.
I was about to say this. I thought alcohol has technically calories that, you know, when people or anyone drinks alcohol, it has calories. Does that participate in a cellular respiration as well?
Absolutely.
I doubt it's not that important for this stage.
No, but it does matter, you know, like anything that has effective effective calories that can actually be used by your body for energy feeds into this process somewhere. All right, which is pretty interesting. So, you know, people will say things like, "Oh, actually, did you know that sugar becomes fat in your body more quickly than fat becomes fat in your body?" And if you hear that as a child, you might be like, well, that doesn't make any sense. Why doesn't your body just take the fat and keep it as fat and then put it into fat? You know, but that's not really how your body operates. The reason that statement does make sense is because you get more energy from the sugars total. And if you have an excess of energy, some of that energy will be used to build up triglycerides. You see what I mean? So that had something to do with this figure. It felt like it was relevant. Was that a satisfying answer to your question?
Yeah.
Okay, I'll take that. I'll accept the lie. Very cool. So hey, it's one of these flow charts. Everybody loves these flow charts, right? Yeah, let's look at it. Let's fill it out together. So cellular respiration generates ATP, which is energy for cellular work, by a process called chemiosmosis, which uses a proton gradient. Boom, there's a full line of reasoning right there. So let's go ahead and look at some of the other steps. Let's look at A. Cellular respiration has 3 stages, all right? What's the first stage? Stage A, stage 1. Glycolysis, which produces some ATP. What is stage 2, indicated by this little spinning symbol? Exactly, and we'll technically say it's pyruvate oxidation plus the Krebs cycle or the citric acid cycle, depending on what you're reading, okay? But that's stage 2, all right? And then stage 3, which produces mini ATP, is called? Exactly, oxidative phosphorylation. Okay. Cellular respiration oxidizes glucose and organic fuels, so it pulls the electrons off of them, and it makes which 2 molecules? Water and CO2. You got it. There's water, there's carbon dioxide. Very cool. So cellular respiration also uses something to pull electrons down, something which pumps protons to create a proton gradient. Now, this one is the tricky one, D right here. So let's hit E, because it's, you know, more approachable. There's even a diagram. What's E representing? The electron transportation, I heard, or the electron transport chain. Exactly. So the electrons go down the electron transport chain to a lower energy state. Now, what is over here pulling electrons down the electron transport chain? Exactly, oxygen. Okay? So cellular respiration is using oxygen to pull electrons down the electron transport chain, because this process only works if you have something very electronegative to pull the electrons away. Cool? Anybody need any refresher on any of these things? Nice, we got 20 minutes left in lecture, and we're gonna get into week 8 material. This is a very exciting time. I'm excited for you, you know? A question. Oh, a question over here? No? All right, fair enough. Question rescinded. You know, since you rescinded the question, I'm gonna have to take those points back, though. Sorry. I've caused discord in the room. Okay, take your points back. All right. Photosynthesis. It's a rainbow. Yay. Hey, there's the other one, right? Can you see it, or is it just a trick of the light for me? There's a second faint rainbow. It's a double rainbow. That's a reference. That's a meme. I'm hip. I'm with it. Have I shown you my move yet? It's very cool. All right. So photosynthesis. We're going to talk about it. It's going to have so many similarities to cellular respiration. That's why I was hitting the electron transport chain and chemiosmosis just so aggressively. It's because there's an electron transport chain plus chemiosmosis going on in photosynthesis as well. So let's introduce the concept. Photosynthesis nourishes almost the entire living world because it generates these high energy organic molecules that then are going to be consumed by other organisms, and that energy sort of flows up the trophic structures, and you'll learn about all of that in your ecology course if you take one, or you probably learned, you did a food, not the pyramid of like, not the nutrition pyramid, but I'm sure you looked at some sort of food pyramid thing in one of your, it's a high school class thing. You know what I'm trying to say to you? Like, I got a dang. Something like this, where you've got like primary producers on the bottom, which are usually illustrated as plants. That's a plant. And then you've got primary consumers on top of them, which are animals that eat the plants, which are used like a bug. There's a little bug. Cute. And then you have secondary, tertiary, and quaternary consumers. And it'll be like, I don't know, a mouse. I draw a mouse, you think? There we go. I don't know what to tell you on this one. That's pretty good. Thanks. And then the tertiary one is like, I don't know, something else. The point is, you've probably seen these trophic structures before in some sort of class. We're at the bottom of the whole process. You've got these primary producers, which are carrying out photosynthesis, generating this biomass that's going to be consumed by all these additional levels. And then these organisms are going to break down the energy in that biomass to make their own biomass and carry out their own activities, allowing them to survive. All right? So bioenergetically, globally, bioenergetically, all right, we've got photosynthesis and cellular respiration just going do-do-do-do-do-do-do-do. All right? Cool. Any questions about this? Is this cool? Solid. So here's the equation for cellular respiration, and here's the equation for photosynthesis. And you'll note that they are basically the opposite of one another. So it really makes this a nice, clear, cyclical process. So whereas cellular respiration breaks down high-energy organic molecules using oxygen as a terminal electron acceptor and generates carbon dioxide, water, and energy, photosynthesis consumes carbon dioxide, water, and energy in order to generate glucose, and it also produces some oxygen. All right? Bless you. And the oxygen here is really sort of a byproduct. But anyway. Any questions about this? Yes, please.
From what I understand, between cellular respiration and photosynthesis, effectively that's the main method that like solar energy manages to circulate into our cycle, right?
Yeah, yep. So you've got this radiation that's blasting the globe, and then that gets fixed into a chemical potential energy. And then by photosynthesizing organisms. And then the rest of the globe is just reliant on eating either that chemical energy and breaking it down, or eating something else that ate that chemical energy and broke it down and now has made its own molecules that have chemical energy. Yeah, awesome. And there are ecosystems that rely on energy sources that aren't solar energy, like in the deep sea. But that's a whole other thing. Okay. Yeah. It's fascinating, but you'll hopefully learn about it someplace else. You know, like the tube worms? You've heard about the tube worms on the thermal vents? It's super cool. Yeah, look them up. Look up tube worms. Safe search off. All right? Sorry. It's tired. It's tired and feeling silly. All right. Photosynthesis. It's sometimes we'll get caught up on like chloroplasts, chlorophyll, all right, and then all the different structures inside a chloroplast. So the first thing we have to do is talk about chloroplasts. Chloroplasts are the sites of photosynthesis in eukaryotes. Because remember, these are membrane-bound organelles. So mitochondria, chloroplasts, those aren't around in the prokaryotic organisms. But in eukaryotic organisms that carry out photosynthesis, chloroplasts are where the photosynthesis happens. Are there examples of species that have maybe not this developed of a chloroplast? 100%. All right? Or some intermediate structure that's not quite a chloroplast? 100%. All right? But we're going to talk about the idealized chloroplast right here. So, chloroplast structure. There's an outer membrane. There's an inner membrane. Between the two is an intermembrane space. Then there's a third membrane inside of the inner membrane called the thylakoid membrane. This is the membrane that's all folded up to maximize surface area. So you know that this is the membrane that's going to have all the essential enzymes and proteins embedded in it for carrying out photosynthesis, right? Because why else would it maximize that surface area of that membrane? This is where protons are going to get pumped across. This is where we're going to have an electron transport chain. The thylakoid membrane is where we're going to have those processes analogous to those that occurred in the inner mitochondrial membrane. And just to say it one last time, that inner mitochondrial membrane is all folded for the same reason, to maximize surface area so you can carry out more of those reactions. I know I say it a lot, just because I want you to leave this class knowing that. Okay, so chloroplasts are the organelle. Chlorophyll is the pigment, okay? Chlorophyll is a molecule that absorbs certain wavelengths of radiation. And there are different types of chlorophyll. You'll be isolating some different pigments in this week's lab, and you're going to isolate some chlorophyll A, and some chlorophyll B, and some carotenoids, and all these different pigments out of spinach leaf. All right? It's going to be a lot of fun. You'll separate them all with thin layer chromatography. It's really cool. Chlorophyll's considered, for teaching, squeaky floor, all right? It's our most important pigment for photosynthesis because it drives the most photosynthesis. Pigments absorb energy, all right? They absorb radiation. It's really possible because of this porphyrin ring structure right here. Look, it's got a magnesium atom in the center of it, and all these nitrogens. Oh, those are pretty reactive things, right? Like nitrogen's super electronegative. This is basically like an antenna, okay, that's going to get blasted with energy, and the electrons in it are going to go woo all over the place, all right? This thing can get supercharged. Now, this long hydrocarbon region is just there. It's nice, it's nonpolar, all right? So this is for embedding, okay? Embedding into hydrophobic domains, like those inside of a membrane, a phospholipid bilayer. Okay? Cool. Technically, these pigments get embedded within a protein, which is itself embedded in the phospholipid bilayer. But in order for the protein to be embedded in the phospholipid bilayer, the protein must be nonpolar, right? So it makes sense that if the pigments are going to be embedded in that protein, they must also have a nice nonpolar region, hydrocarbon, carbon-hydrogen bonds. Okay, chloroplasts, here's like a standard issue. It's a cross-section of a leaf, okay? Look, showing you the mesophyll cells, of the leaf where all these chloroplasts are sort of concentrated, okay? Fascinating stuff. This also shows you stomata, which you'll learn about in Botany 105, which probably all of you need to take or have taken already. No, yes, no, no? Okay. Hey, listen, this is really interesting stuff, the stomata. Plants don't have lungs. We've got, this big empty cavity inside of our body, and we just move this diaphragm up and down that like makes a vacuum and sucks air into our lungs so that we can get gases to exchange really quickly. Plants don't have lungs, all right? Plants have these little holes in their leaves, all right, that are covered by these guard cells. And when the guard cells swell up with water, it opens the hole, and it just lets oxygen diffuse out and carbon dioxide diffuse in. So this is how the plants get the carbon dioxide that they need in order to carry out photosynthesis. This is also going to have the consequence of it's like literally opening up a hole on your arm. The material inside is going to dry out pretty quickly. And that's one of the consequences of stomatal opening. This is the primary path by which most plants lose water, and stomatal regulation is a whole field of study, and it's all very fascinating, and it's outside the scope of our BIOL 105 class. All right? But I can't help myself but talk about it a little bit. So I'm going to use this to tie it into something relevant, which is, remember, right? Photosynthesis is going to consume CO2 and produce oxygen. So if you open a stomata, the concentration of carbon dioxide inside of the leaf will be lower than outside of the leaf because you've been consuming it, and the concentration of oxygen will be higher inside the leaf than outside the leaf because you've been producing it. So the gases are just going to move by diffusion, by the rules of diffusion. Cool? I love it. Hey, check it out. It's a redox process. That means that some things get reduced and some things get oxidized. All right? Now, in cellular respiration, glucose got oxidized, remember? In cellular respiration, glucose got oxidized. The electrons got pulled off of it. So in order to build some glucose, you have to reduce something else, throw some electrons onto something. And the electrons are going to get thrown onto carbon dioxide. Okay? So this one's a little bit tricky because of the point here where water gets oxidized. It doesn't make a ton of sense just looking at this figure, at this slide here. Because the one thing we know about oxygen is it's super electronegative. It's the only thing we've talked about with oxygen, all right? So it's going to be really, really hard to oxidize water to release that oxygen, to get those electrons. But plants and the process of photosynthesis have the energy of the sun, okay? So the plant is just going to take the energy from the sun, all that radiation, and sort of focus it via all of these different pigments, which are all going to vibrate and zap energy into this focal point. And all that energy is going to be used to blast the electrons off of water, oxidizing the water. Now, that's an oversimplification because the energy is actually going to be used to blast electrons off of a different oxidizing agent, which then is going to pull the electrons off of water. But I'm trying to just lay the groundwork of how is it possible that you're going to oxidize water? That should take a ton of energy. And it's like, yeah, it does. It takes the power of the sun. Okay, photosynthesis is an endergonic process. High energy products, low energy reactants. Cool? I love it. Photosynthesis is driven by radiation. Radiation within the electromagnetic spectrum. The electromagnetic spectrum spans a bunch of different types of radiation, including things like gamma rays, X-rays, ultraviolet radiation, infrared radiation, microwaves, and radio waves. All of these are defined based on the wavelength of the radiation. So if it's got a long wavelength, it's low energy. If it's got a short wavelength, it's high energy, right? We cool with all that stuff? And here in the middle of all those different types of radiation are the wavelengths of light or wavelengths of electromagnetic radiation that we can see that we call visible light. All right, the spectrum of light. All right, here we go. It runs from 380 to 750 nanometers in wavelength. The shortest wavelengths have the highest energy. The longest wavelengths have the lowest energy. Oh, hey, don't get too much blue light right before bed. Oh, that's interesting. It's higher energy than red light. Huh. Infrared radiation. You never hear about that being dangerous. In fact, you'll go into an infrared sauna pod, maybe. I don't know if you've ever done that. I've never done it, but I see them advertising them. All right. You're all emitting infrared radiation right now. That's just heat radiation. It's very low energy. Okay? Ultraviolet radiation is very high energy. It's higher energy than anything in the visible spectrum. And you get too much UV light, all right, it can cause problems because it's powerful enough to penetrate into your cells, maybe get through your nucleus, maybe disrupt the structure of your DNA, give you a dang old melanoma or something, and you don't want that, all right? You also don't want too many X-rays hitting you. That's why they wear the vest at the dentist, all right? And you don't want gamma rays because you turn into a Hulk, okay? All right, cool. So radiation, this is all radiation. The phrase radiation encapsulates all of these things, not all of which are necessarily harmful to you as a person. So we're just going to kind of destigmatize some electromagnetic radiation and understand more of the nuance to it. All right, that'd be a bad thing to say. Here we go. The plant absorbs specific wavelengths of light, and the wavelengths of light that it absorbs determine its color, all right? But really, the color of the plant is green. The color of photosynthetic material is green because the pigments absorb all of the wavelengths besides green wavelengths of light. That's a really key thing to remember, all right? If you've got a green sweatshirt on, then the pigments there reflect green light, okay? They do not absorb green light. I really want you to, I know that might seem like, oh, we know this stuff, but you're going to be generating these sorts of figures in lab this week, all right? So I want you to be kind of comfortable with the idea of, hey, check this out. These pigments do not absorb green light. That's why the pigments appear to be green. The pigments absorb these other wavelengths of light, all right? Red, purple, blue wavelengths, and that gives you the color. Carotenoids, this class of pigment, they appear orange because they absorb these wavelengths of light, but not green and red light. So they reflect yellow and red light. And so they appear orange, carotenoid, like a carrot, all right? That I have used up. Oh, I got time for like a quick question. Yeah, go.
What about purple plants?
They have anthocyanins, it's a different pigment. It reflects purple light, absorbs other wavelengths of light.
But it's photosynthesis still.
Anthocyanins are usually not involved in photosynthesis to the same degree, but it's complicated.
That's why the leaves are so
Yeah, they'll still be green. Yeah, usually floral pigments are entirely for pollinator attraction. Leaf petals don't usually, or flower petals don't usually carry out photosynthesis, except for like at a very, very limited level. Cool. Have a great day, everybody. Wonderful Wednesday. I'll see you again on Friday. Have a good lab. I'm going to stop this recording and I can answer any questions.